Okay, I will switch off the this one. Okay.
Okay. So good morning, everyone. It's my great pleasure to host Professor J. Kizhakkedathu from University of British Columbia with us. I do apologize for minor technical issues that we had before we started the meeting. For those of you who have been with us like for 2 years, you've probably witnessed some of these technical issues before, and that's sometimes due to the challenges we have with the app that we are using at the moment. But nevertheless, it's awesome to have an expert in the field of biomaterials with us today. I'm very excited to have Dr. Kizhakkedathu with us, who will be talking about infection-resistant coatings for basically fighting bacterial resistance. This is a very important topic that basically infectious diseases, and then also in the realm of diseases such as COVID. These kinds of materials have many applications in the field. So Dr. Kizhakkedathu is currently a professor at the Department of Pathology and Laboratory Medicine at the University of British Columbia. He is also a Michael Smith Foundation for Health Research scholar, and then associate member of Department of Chemistry. He has achieved he has many achievements. I don't want to get into all of those given the limited time that Dr. Kizhakkedathu has today. I just wanted to directly go into his talk, and then we'll give him the opportunity to talk about his research in the area of infection-resistant coatings. So without further ado, I invite Dr. Kizhakkedathu to start his presentation, and then during the talk, if you have any questions, feel free to use the Ask a Question box. Share your thoughts and questions with our speaker. Thank you very much. Dr. Kizhakkedathu, the floor is yours now.
Okay. Thank you very much. Can you hear me well?
I can hear you loud and clear, and your slides are well on the screen. Thank you very much.
Okay.
Greatly.
Thank you. Good morning, everyone. And sorry about the trouble, actually. So these technical troubles are, always a challenge. So welcome to today's talk on infection-resistant coating. And I would like to thank Dr. Kari for the invitation and the audience for listening to. So today, my agenda is kind of like give you some introduction to the work we've been doing on last several years, and this is a part of our the work in the lab, and you'll go from there. And so before beginning the talk, I would like to disclose today's talk actually I'll not be talking any of those companies, any of those involved in these companies. So let's begin talking about kind of like my lab at the British Columbia, looking at we are interested in looking at developing molecules and materials for blood-related applications, and we have a very vibrant program looking at 4 different areas. We are looking at including macromolecular therapeutics, proteomic reagents, and cell surface engineering. Today, I'll be mainly looking at kind of this one area where I'll highlight this blood-compatible surfaces and coating is actually we are looking at. Today, I'll give you some examples of kind of like the surface coatings actually, and currently actually we are moving towards developing these coatings for blood-compatible device applications. So today's agenda, I'll introduce you to some of the device-associated or device surface modifications, and then I will introduce you to how anti-fouling or anti-biofilm coatings are kind of important, and what are the criteria for developing these coatings. And then also looking at kind of like, what are the new approaches actually we took to develop this. And then finally, actually, like looking at in each of those examples, I'll show you some the mouse models or pig model studies to show that its activity in a complex environment. So why we need to develop this kind of surfaces? Actually, if you look at the current clinical scenario, implant-associated or nosocomial infections are kind of like a major challenge, and implant and device-associated infections is majority of these hospital-acquired infections. And some of those devices actually which are very prone to infections are such as urinary catheters, vascular devices, and blood storage devices are kind of like some of the challenges. And if you look at these are kind of like a major problem due to the fact that actually there is a prevalence of multi-drug resistant bacteria, and because of that, actually treating these patients are very, very challenging actually. So can we develop actually better coding or a kind of a device actually which can resist the infection by its own. Actually, that's a kind of a good scientific problem to address. And another challenge is kind of like we have the antibiotics. Actually, there are hundreds of there are many antibiotics are available, but these antibiotics actually when it's attached to the surface or attached to a device or a surface, it is most often actually become ineffective and actually it's not going to work. And so then we need a kind of a different methods to treat infection associated with the devices. And another aspect is actually like you have kind of like it can develop kind of like antimicrobial surface, and is it be biocompatible? So you have to work around this kind of like two aspects to understand actually what is happening. And another challenge is kind of like treating biofilms. Actually, most often the biofilms are very is a different form of colonies of bacteria, and this bacteria actually like is very resistant to antibiotics, and often like almost like 1,000-fold higher concentration of the antibiotics are needed to treat the biofilm. So all of these kind of like complications are actually like are challenging, and also like these challenges one has to overcome. And these are kind of like some of the examples of like devices and implants actually I already talked about. Actually, like for example, vascular catheters, urinary catheters, and blood bags, and then the vascular devices. You could see that actually like the surfaces or these devices are kind of placed in kind of different parts of the body, and the challenges associated with actually at that places where can it be biocompatible, or whether it is active in that particular location, and these are also another challenge one has to face. So how can we achieve, or how can we develop a better biocompatible antimicrobial and biofilm surfaces? And what we have seen actually, the literature shows that actually like if you could have kind of like a covalent coating or covalent surface coating, and it is going to be kind of like a stable, and also kind of like a hydrophilic surface coating actually, or antifouling surface coating could be one of the criteria people has to when the researchers has to think about developing a surface coating. Although the different applications or the different locations actually these are placed, but anti-adhesive coatings are always have advantages there. And this kind of coatings actually, when one could we could think about kind of like a protein resistance or bacterial adhesion resistant surfaces are most often actually useful to prevent further biofilm formation for the bacterial infection colonization happening. Due to the fact that actually although antimicrobial surfaces can kill the bacteria, but the dead bacteria can accumulate on the surface, and most often actually like that surface becoming ineffective during the time, and then the surface becoming again get colonized. So it's you don't have kind of like a continuous or long-term activity. And another challenge or another criteria is kind of like, if a coating can be developed actually, is it applicable to diverse substrate or diverse surfaces? Biomedical, implants or devices are made of plastics, metals, and ceramics, and different compositions and different surface chemistry. So is it applicable to diverse substrates? And then another kind of thing is actually like, is it coatings or is kind of like coatings applicable to diverse antimicrobial agents, or is it biocompatible? And all of those, and you need to have kind of broad spectrum activity as well as long-term activity in complex conditions. So these are the kind of like, a criteria actually one has to think about kind of developing better surfaces or better antibiofilm surfaces. So in our early work, actually, like, started in 2009, 2010 time, and we looked at, we were working on kind of like a polymer layers or polymer surface coating, actually, which is called polymer brushes, and surface anchored polymers. And we thought, okay, can we adapt this kind of coatings to developing antimicrobial surfaces? Our goal is kind of like using a functionalized polymer layer to attach antimicrobial peptides or antimicrobial agent on the surface, and to generate kind of like a surface which is really antimicrobial as well as anti-adhesive actually. So these polymer brushes properties do have kind of like, it provided anti-adhesive properties. And then if you attach the right type of antimicrobial agent, in this case, actually, we were using antimicrobial peptide. The reason being, actually, like, this has got a multi-mode of action, and then it could, most often, actually, like, there is less chance of developing drug resistance in this case. So we, using this concept, actually, like, we started from in an example using a titanium surface. We used a surface initiated polymerization to generate a surface grafted polymer, and then we modified with a peptide linking agent, and then utilizing a cysteine containing peptides to generate a peptide coated or peptide conjugated surface. And the most important aspect is actually we could change the polymer, and we could change kind of the length of the polymer. That means actually how much peptides can be attached, and also actually we could change the peptides as needed. And antimicrobial peptides, actually, diverse range of antimicrobial peptides are available. So this is one example of those, actually. So you could, you have kind of like a variety of peptides are available. So you could utilize this concept to generate a surface coating, actually, which are really anti-adhesive as well as antimicrobial, actually. So we could we have versatility to change diverse properties of these two for different applications. So after looking at, actually, after developing this, we looked at this activity, and one example is shown here. Actually, this is on Pseudomonas aeruginosa. So actually, you could see that the peptide coated is effective in killing the bug, and it can kill gram-positive bacteria too, as well as gram-negative Pseudomonas, and it is very effective in killing the bacteria. And then we moved further at that time, looking at, actually, it's implanted in a subcutaneous implantation in a rat, and we utilize kind of like the right side. We immobilized the peptide coated implant, and the left side, actually, we immobilized, we implanted the control, and then we instead the bacteria there to see whether it's effective. And what we have seen that, actually, like, these coated implants are highly effective in preventing the infection in these complex conditions. And these are some of the first studies, actually, to show that, actually, that conjugated peptides are in an antifouling surface is kind of very effective as a tool to develop implant infection resistant surfaces for devices and implants. So moving further, we kind of like this chemistry is adaptable to diverse substrates, but although actually need to have kind of like multiple modification. So in the case of we adopted this method to a urinary catheter. Actually, like earlier it was a titanium implant, and here actually we have a plastic implant. Here urinary catheter, we utilize kind of like a plasma modification to, and followed by the surface initiated polymerization to generate this peptide coated urinary catheter, and then we further utilized a urinary tract infection model in association with the Dr. Lang at UBC. So here actually we kind of like we modified a catheter, and then a piece of the catheter is implanted in the bladder of the mice, and then we after placing this implant this piece of the catheter, we challenged with the bacteria, and then after 7 days actually looked at how much bacteria is present, or whether whether this implant is able to, whether this the device is able to kill the bacteria. So using this model, and we looked at kind of this activity of this surface, and as you can see that on the left side, the catheter is able to kill the bacteria very effectively. Almost 5 log reduction is achieved here. And it's also kind of effective in killing, this coating is antimicrobial, is affecting killing the planktonic growth. Actually, what is growing in the urine actually is also able to kill that. So it is kind of like it is very, very gratifying to see that actually, like from a concept to all the way to kind of like using in a mouse model to show a very effective surface coating actually it could kill that. So one of the challenge what we faced, although we learned a significant amount of kind of like understanding on what are the criteria for developing the better antimicrobial surface, and what are the properties actually needed. But one of the challenges here is actually like you need multiple steps of modification, and which makes it the translation of this kind of coatings are really challenging. So for example, a urinary catheter actually like is kind of like under $10, and if you want to do is kind of like this kind of like multiple modifications, although it is is going to be effective, but the price is not going to be kind of like it's not a useful criteria, so that actually like it could translate into commercial applications. So then what we thought about actually, is there any other method? We learned a lot from this work, but can we develop a better coding actually which is easy to modify, which is easy to translate? And that's what I'm going to talk about the next section. So we using that principles actually we learned, and we came up with another universal anti-adhesive coating actually, which is where actually like it is a very simple process actually we came up with. So we mixed 2 different polymers actually, like one is a monomer and high molecular weight polymer actually, like we mix together and in situ polymerize to generate a coating, and which can be coated on the surface using either a coating or kind of a spray coating or any other coating, those kind of method. It's a really remarkable method in a way that actually could coat any surface within from few minutes to a couple of hours, and we can generate a better coating. So how we develop this coating is the fact that actually like we screened in combination with the dopamine as a universal binder, and utilized kind of like a multitude of different polymers actually structures. So these polymers are shown here, and so we screened these polymers in combinations to identify what kind of like what are the properties, or what are the what type of polymers are going to be effective in generating a better coating. So what we have seen initially, we started from an NDX format, and we generated a coating on the surface, and then it seeded bacteria, and we looked at after 24 hours actually how much bacteria is adhering, and our criteria is that what are the surface coating actually generate the lowest, the bacterial adhesion, and we could move forward to the next set of analysis. So in this coding process, or this screening process, actually we identified the fact that we could utilized low molecular weight polymers, as well as high molecular weight polymers, as well as ultra high molecular weight polymers. And what we found that actually, some of the ultra high molecular weights, such as PDMA, I'll show a polydimethyl acrylamide, you could see that there is very little amount of bacterial adhesion is happening in combination in this binary combination coding. So we looked at, we identified that actually the polydimethyl acrylamide was one of the good candidate, and then we looked at changing the molecular weight of that, and then what we found, the molecular weights in the range of closer to 800,000 to 1 million Dalton, and it could, in combination with the dopamine, we were able to generate kind of a coding which is very resistant to biofilm, formation. And what we have found that actually this particular coding, and it can be this coding can be applied to the surface by a deep coding process or a spray coding process, and also it is applicable to kind of a diverse substrate. It is universally applicable to many different substrates, and also kind of we could see the example shown here on different substrates, and the bacterial adhesion is kind of almost kind of prevented here. And we further looked at kind of different types of bacteria, Staph aureus, MRSA, E. coli, and in this example, and also we have seen actually it's a very thin coding. And remarkably, actually, what we have seen, and this particular coding is very effective in preventing the biofilm formation over long periods. And this is an example shown here is in a flow condition. And every day, we challenge this, the modified surface in a flow loop, and every day we change the bacteria culture, and in a flow conditions, and it can see on the top. You can the cursor. This is control substrate or substrate. And this substrate you can see that actually after 28 days of continuous exposure to the bacteria and bacteria culture, you don't see kind of biofilm or very minimal biofilm formation or bacterial adhesion is happening here. And we calculated the biofilm, and that is it's a cyto green kind of labeled bacteria there, or biofilm there. And on the right side, you could see that this is the biomass deposited. You could see that almost 98 to 99% kind of decrease in the biofilm deposition over 28 days, which is kind of one of the kind of at that time, we published this on 2018, and it's at that time this is kind of a benchmark for kind of showing our 28 days actually it could prevent the bacterial adhesion over the long term. And this is further proven by scanning electron microscope graph. You can shown here on the bottom. This is encoded. You can see that actually carpet of bacteria or biofilm is there, and on the right side you can see that the coded substrate is kind of able to prevent the adhesion of the bacteria. And further, we looked at kind of whether this although this is good in vitro, whether it is going to be effective in vivo. Actually, that is what we shown in utilizing kind of the Lang's model. Dr. Lang actually helped us to evaluate this and use this mouse model of infection or urinary catheter infection, looking at kind of its activity. Actually, you can see that the surface, the coded surface, is able to prevent the bacterial adhesion remarkably well, but since this coding is not effective it does not have any antimicrobial agent, it is not able to kill the bacteria in the urine, and that is kind of this work shows actually prevent, or it could using kind of like anti-fouling surface, you are effective in you'll be effective in it could effectively prevent the biofilm formation on the surface. But what about kind of like if you have an already infected a place where we want to place a device, or for example, actually like a patient actually already have a urinary catheter infection, but if you want to replace and then put a new catheter, and then can we utilize this kind of approaches to use in that kind of environment where you might need kind of like an antimicrobial agent, which I'll come to that. So we further utilize this concept in looking at kind of in a pig model of urinary catheter infection. You could see here a pig the catheter has, modification has been adapted to a large catheter size, and you could see that actually like almost 95 to 97% the prevention of like biofilm formation is achieved here, and in here, so we place a catheter, we kind of like instill the bacteria and waited for 7 days and remove the catheter and analyze the bacteria on the surface. And this studies shown that actually going from the concept to all the way to big studies shown that actually like this kind of coatings are really effective in preventing the biofilm formation. As I said actually earlier, actually like this surface coated actually like surface coating doesn't have any antimicrobial agent. And if you can add an additional antimicrobial agent to that actually, so you have an anti-fouling coating plus an antimicrobial agent actually, as I mentioned in the early part of my talk, and this will be remarkably good actually. So here, then we further modified, we developed the chemistry to attach an antimicrobial peptides on the surface, and still able to kind of like prevent the bacterial adhesion, as well as it is able to kill the bacteria, and then we looked at that. So here actually like we further utilize this our 96 format, and we screened kind of like number of peptides and number of bacteria, and we identified kind of like a set of peptides actually which are in combinations with this antimicrobe, the anti-fouling surface, which is remarkably in preventing the biofilm formation. So we further looked at infection model in mice, as you can see that, and with the addition of kind of like the antimicrobial agent, the further reduction of kind of like the biofilm formation, as well as actually it could kill the bacteria on the surface. Bacteria in the planktonic medium, which is kind of like a combination device where actually like you'll be able to take this to a patient actually which is already infected. So this so far actually like how talked about kind of like an antimicrobial peptides as well as anti-fouling surface. So can we utilize this kind of like concept in another setting actually? So can we utilize in another kind of antimicrobial agent actually which are usable actually, which are also kind of like important in does not have kind of like a prevalence of drug resistance or kind of like drug resistant properties. So they're actually like one of the silver is one of the most usable or kind of like most important antimicrobial agent, and it is used extensively from the last 100 years or so in treating infection. But one of the challenges for the silver, although it is remarkable that it's very useful, is the toxicity is kind of like one of the challenges for in the case of surface as well as in treating using silver. And another challenge with the silver is its release property is kind of like not ideal, and it has to be kind of like it has to be not always kind of like you can control the release. So we thought actually like the concept actually we achieved so far on the anti-fouling as well as attaching antimicrobial agent can be utilized to adapt it to a silver to generate a silver containing coating, and which is going to be useful for preventing the infection. So here we came up with kind of like a combination or kind of like a composite coating, and which is of kind of like 4 different components, including a hydrophilic polymer, a cross-linker, and as a binder, and as well as silver, in combination together to generate a unique composition, actually, which can be applicable to the surface. So what we looked at to identify that composition, we used a high throughput screening, and we found that actually is this 4-component system in combination with everything. And we screened almost kind of like 168 or about 175 different compositions. And from there, actually, we identify a unique composition with containing a hydrophilic polymer, and here again the PDMA, and which is kind of like which is able to kill the bacteria. And the screening is done for 24 hours, and then we further extend the screening for kind of like 7 days. And what we found that actually like molecular weight as well as the composition of these coatings are really important in generating kind of a coating which is kind of like long-term activity. So our criteria for kind of like generating this coating, just kind of the screening process is kind of like extended over 7 days. The reason being is actually like we want to make sure that actually like this surface coating it do have kind of the long-term activity. So we screened this coating over 4 weeks, and with the continuous exposure to bacteria over 8 different types of bacteria. I could see that our coating is able to, the green color is for the live bacteria, and red color is for the dead bacteria. And our coating actually does not, it prevent the bacteria adhesion over a long period of time. And it is, it's remarkable to see that actually over 4 weeks, actually you could see the bacterial adhesion is almost null there. And remarkably, actually, it could also kill the bacteria in the solution, actually. So that means, it could kill on, bacteria adhering on the surface, as well as actually like it is free floating bacteria. So it could be used for an infection, treating in infected device can be treated there. And we have seen that actually like in challenging conditions too. And this safe coating is kind of like, it is remarkable in preventing the adhesion. And here again, the controlled silver coating actually could, by 28 days, so like you could see that like a lot of biofilm information. And as I mentioned earlier, actually this is to the fact that dead bacteria getting adhered to the surface, and again, and on that actually the bacteria colonization is happening. But these anti-adhesive properties in combination with the silver, this is able to kind of like prevent the bacteria, the adhesion of that. And we looked at further in an implantation model to look at its activity. And here the titanium implant is implanted, and instead with bacteria, you could see that several orders of magnitude decrease in the in the bacteria count. And also this surface is kind of like really biocompatible. Actually, that is one of the challenge for the silver. And what we found that is coating placed in kind of like in this kind of like hydrophilic environment, and you can make it kind of like really biocompatible and non-toxic. And the reason being is actually like we were able to achieve the control release of the silver, and also kind of like this silver is kind of like placed in kind of like in situ formed silver nanoparticles are used here, and that makes it kind of like this control release is possible there. And we could also kind of like this coating is applicable to diverse different substrate or different property, diverse different shapes, and also is very effective in all of those conditions. And it's also kind of like very lubricious, and it has got high mechanical properties, and it is abrasion resistant, and also kind of like it is very stable using kind of like many different methods, actually like mechanical challenges actually we put it there. And so we again actually looked at this coding, and it is in infection model. Like you could see that this is able to kind of kill the bacteria in a mouse catheter model, catheter associated infection model, and it is very very effective. So where we are going from here, and we are further looking at actually this coding. We could see that actually this coding can be applicable to kind of adaptable to large catheters in a single step, kind of within our coding. Actually, we could translate a infection resistant catheter, a human-sized catheter, actually without much challenges, unlike the polymer brush coding. So we are further moving ahead with this kind of big studies, and other studies are ongoing, and we'll be able to talk to you actually those kind of things in the future. So in conclusion, actually what I have shown today is, although I had to go a little faster to finish, and in conclusion, what I have shown here actually. So what what are the criteria for generating an antibiofilm coding, actually, which starting from kind of a polymer layer, and then we attaching antimicrobial peptides. Actually, we were able to kind of arrive at certain criteria, and showing that actually the excellent coding can be developed. And because actually we are using kind of antimicrobial peptides as well as silver, the chances of developing resistance is going to be really low here, actually. So I won't discount that actually there might be a chance, but it is really low here. And the polymer chemistry part is kind of an important aspect, and and the screening of diverse polymers, and the finding out the right molecular weight, and what kind of density of the functionality there, and all of those kind of things are important in generating this coding. And most remarkably, kind of the anti-adhesive nature of the coding is one of the most important aspect, in combination of anti-adhesive nature of the coding plus the antimicrobial agent, is able to provide kind of this long-term activity. And this coding, what we have developed is a substrate-independent coding, and it can be applicable to diverse substrates. And what I have shown also kind of this, not only actually these codings are effective in vitro in normal in the lab, but it is also effective in a mouse model as well as in pig model, and it is so it has got kind of a good translation potential. And the coding is also kind of highly biocompatible, and in many aspects, actually, we looked at it is really non-toxic, And before concluding, I would like to thank my group. Consists of very talented postdocs and graduate students, actually, which are they did this work, especially and other Hussein and other people actually who have contributed this kind of antimicrobial aspect. And in my collaborators, especially Dr. Lang, who developed this mouse model as well as a pig model, and Hancock, actually he has developed antimicrobial peptides, and other people actually who are Greco, Wang, and Strauss. Susan actually helped in many aspects of this project, and also kind of finding agencies who supported this work. And thank you very much for listening. And I had to go a little faster, and because of the technical difficulties, I had to we started late, and I would be happy to answer any questions you might have. And thank you very much.
Thank you very much, Dr. Kizhakkedathu, for your excellent presentation. Can you hear me?
Yeah, I can hear you.
Wonderful talk, and this is an area that I'm very interested in, because we're doing some research in the area of wound management, and infection is a major challenge in wound management, as you may know. But But I encourage the participants to ask questions if they have any using the Ask a Question box. I have many questions here, and I don't know how much time you have, Dr. Kizhakkedathu, but just let me know when I have to stop, and then I'll stop asking you questions, because I'm very excited about this topic, and then I'd like to.
I'll have 5 minutes. I have to leave by 10 o'clock. Okay.
Okay, that's great. That's okay. So then I'll ask the most important question. So so this is a very cool technology. So have you? I mean, you use this for mostly for medical applications. Have you thought about using this technology for example, for food packaging, or for as anti-fouling agents for marine applications as well? Or...
Yeah. Can I? Yeah. How thank you very much, Rosen, for that question. Actually. So we have medical applications like it's more challenging actually. This so what we have seen actually this kind of the food packaging as well as marine applications and all. But the challenge there is actually the high volume of the work actually, whether it can be able to, in the case of antimicrobial peptides, is going to be a costly technology. So the food packaging, whether it's going to be useful there. So whether it can be accommodated in that price range. But in the marine environment, it is maybe a little more complicated than that, actually, in a paint or other kind of system, whether we could utilize that. So there, we may not need to develop this kind of a high-end technology for that. But I would say that actually this anti-fouling coating for sure, it is really economic. If somebody is interested, actually we'll be able to work on the packaging and all of those kind of things for sure. And we have the polymeric systems, which is it's really economic, and then we can work on. And that is one of the reason actually why we didn't go in that front, but there is a lot of application out there actually is going to be useful.
Wonderful. And the next question is about bacteria. You talked about bacteria. How about fungi?
Yeah. This the peptides. Some of the peptides are kind of it's effective against fungi and also viruses too. But the latest technology, actually, the silver one, actually, which is recently published, and which we have shown that actually it could kill all bacteria and fungi as well as the viruses. Okay, so it is kind of a really remarkable, a coating actually, which is applicable in diverse range. Actually, we have an upcoming publication coming on the antiviral activities of this too, and it's going to come out soon too. So.
Okay, awesome. So I have 2 minutes to ask my last question. So the next question I had was, you mentioned that it's an approach that is basically can be used on many different surfaces. Have you ever tried this method on hydrogels?
Yeah, it works on hydrogels too. Yeah.
Okay, cool. Again, I have so many questions. I'll probably have to ask you at some another point. We're close by, so I'll drop by your office or set up another meeting and ask you these questions. And since, in the interest of time, I would like to thank you again, Dr. Kizhakkedathu, for your very interesting talk. I would also like to thank the participants for joining this event. I apologize for the technical issue we had at the beginning, but we managed to fix it, and then we enjoyed this excellent talk. So with that, I'd like to end this session and then wish you a great rest of the day. Thank you very much.
Thank you very much. Okay, bye-bye.
Bye.